For this next session of the Covering Rare Diseases Fellowship Program, we're exploring the importance of gene therapy for rare disorders and some of the key development challenges to making that progress. We're joined by Dr. Peter Marks. He's the director of the Center for Biologics Evaluation and Research at the Food and Drug Administration. Dr. Marks, thank you so much for joining us today.
My pleasure. Thanks very much for having me. So I have a presentation that probably go about for about 20-25 minutes, and I think there should be time for questions if that's okay. So I thought that I would start by kind of describing the need for rare disease products in the gene therapy end of things. You probably have already heard about that, but I'll tell you from our perspective a little bit about the development and approval of these products, some of the challenges in product development, and some of our efforts at FDA to facilitate product development and some of our pathways or programs that help do that. So in terms of approved gene therapies, we now have 10 gene therapies or cell-based gene therapies in the United States that have been approved. When you look at this list, 6 of these are chimeric antigen receptor T cells. 2 of these are stem cells that are genetically modified and given as part of a stem cell transplant type program. And 2 of them are directly administered gene therapies, one that's locally administered into the eye for a type of hereditary blindness, and the other, which I'll tell you a little bit more about, that's given systemically for a neuromuscular disorder that takes children's lives early in life. And so the one thing I would say is, although there are just 10 products there, there is a very robust global pipeline of products, including many more genetically modified T cells, both ones that are autologous and ones that are allogeneic. Those are generally for, again, relatively uncommon cancers, things that might be considered rare. For hemoglobinopathies, some of the hemoglobinopathies aren't that rare, but they are also in development. Hemophilia A and B, lysosomal storage disorders, which indeed are rare disorders, neuromuscular diseases, any number of which are pretty rare diseases, and the retinal disorders, and a variety of others. I want to say a few more words about kind of the poster child for success of what gene therapy can look like when it really works. Anasemnogene is a gene therapy for one of the more common rare diseases in this space, spinal muscular atrophy. And spinal muscular atrophy comes in various types, but type 1 is a very severe type that takes children's lives, usually by the time they're about 3 years of age. They are born, they look normal, but they're not able to make any of the normal developmental milestones like holding up their heads or sitting up. And eventually they end up on a ventilator and die of complications. There was an oligonucleotide-based therapy that was developed for this, but it had to be given repeatedly. And around the same time, a gene therapy was developed, and it's a one-time intravenously administered gene therapy that is given early in life, usually within the first 6 months of life. When this was investigated in clinical trials, the graphic on the left is just to show you that that dotted line that's going horizontally, below it you're abnormal, above it you're normal. And you can see that early in life, children who received this gene therapy, 14 out of 15 ended up developing normally. The child on your right is this is not where you don't need statistics, you don't need small metrics, because you don't see 3-year-olds with spinal muscular atrophy type 1 that are alive, let alone running around generally. So this is it would be rare to see them alive, and to see them running around is unheard of unless you had something like this intervention. So this brings up the idea that, you know, when it really works, it works well, but there are actually thousands of rare hereditary and acquired diseases that affect anywhere from a few to a few hundred to perhaps a few thousand individuals, and these could be addressed right now potentially with gene therapies because they're single gene disorders. There obviously, though, are a variety of hurdles to getting there, and that's what I think we'll talk a little bit more about. And another thing that's really important is that when we think about rare disease gene therapy is the proving ground for getting it right so that we could ultimately look to gene therapies for larger populations. Because if we can get it right in a lot of different small populations, we'll have more confidence that we can get it right in larger populations. And the gene therapy that we're thinking about here really is a move from personalized medicine, where one looked at biomarkers in individuals and then looked at drugs that one had on a shelf. And instead now moving towards individualized medicine, where one looks at what the problem the individual has and then develops a product to address that, or finds a product that's been developed to address that. And this individualized medicine really breaks down into two large buckets. One are customized products. Those are such as the CAR T cells, where the products have the same indication, the same mode of action, but they have some personalized nature to them. For instance, the autologous CAR T cells, they are harvested from an individual and then modified and given back to that individual. And then there are created products like the gene therapies, where one actually is making products to handle these different gene defects. Now, one could say, why have this kind of, what is this different from our normal paradigm? The difference here is that for these created products, we're often reusing part of the product again and again. That is the gene therapy vector, which then shuttles the gene therapy insert into the patient or the individual. That has some special qualities to it that may allow us to get these therapies developed a little more rapidly by using some tools that I'll tell you about in a few minutes. This is just the difference here between personalized medicine and individualized medicine. You know, in general, what you're looking here is individualized medicine may be broken down into this customized and created. Customized is more like when you have these suits where you can made to measure, as opposed to truly bespoke tailoring, which are what some of these bespoke gene therapies are, where you're really making them for small populations. So just to say what we have at FDA, we in terms of what do we care about this? Well, we do care very much because we are very much involved in the product development ecosystem, and we're responsible for ensuring that the products that come through this system are safe and they meet a legal standard for efficacy. And you have obviously all heard a lot about the tension between either getting access to products too soon in the absence of enough safety and efficacy data, or waiting too late and having patients wait too long and suffer needlessly. And there is a balance. There's not a right or wrong answer. It's simply a balance between those two that we have to find. And so we are involved in the process of product development from conception often through post-marketing in order to work with sponsors who are developing products to bring things through the process. And I'll try to point out to you this balance. Our goal, though, at least I will tell you a philosophy up front here, that our goal here in bringing these rare disease gene therapies forward is to make sure that they do meet our standard for efficacy and that they are safe, so that when patients get these, they can be assured that they're going to bring benefit. Because if that's not the case, we're not doing them a favor. In addition, we're not going to be able to actually get them paid for, because payers will insist that these relatively expensive therapies actually do have a benefit. So when we think about this, we're often doing benefit risk assessments, which means that we have to understand the conditions we're dealing with. We have to understand the treatment options for those conditions, the potential benefits, risks, and all of the uncertainties that go around that. when we go to make these available, ultimately the goal is to get a biologics license application for a gene therapy. While they're under study, they are under investigation new drug applications, but sometimes we make these products available through expanded access provisions for individuals who might not be eligible for clinical trials. You might have heard about right to try. That's when one has a product that meets certain qualifications in terms of its stage of development, where it can be used completely independent of FDA, essentially, if a manufacturer will provide it. It's not been a very large pathway used in this area. I mentioned it. It has issues that we can talk about later if someone wants. Emergency use authorization really is not applicable to gene therapy products in general, but I just put it up here for access. Our ways of promoting product development at FDA, though, are to use any number of levers that are at our grasp. Many of them have come to us through our user fee acts. Now, the Prescription Drug User Fee Act, Medical Device User Fee Act, et cetera, biologics and generics, they all are commitment letters, which are our performance agreements with industry. But when they are passed, those commitment letters, with the exception perhaps of this most recent, there's always an exception to the rule, this most recent batch that was of the most recent PDUFA letter that was came through Congress. Most of the time they come through with what we call the PDUFA Christmas tree of other things that Congress wants us to do to help expedite product development. And in the past, those have included things such as having patient-focused drug development meetings, issuing guidance to help sponsors develop products more rapidly. We also have our orphan designation, priority review vouchers, and expedited development programs. I'm going to tell you a little bit more about those. I already spoke a little bit about the user fee acts and how those, they're reauthorized every 5 years, and they tend to give us more mandates. The orphan designation is something that has been around now for several decades. It was designed to help address rare diseases by giving certain tax credits and exclusivity if certain conditions were met, for conditions with under 200,000 people in the United States affected. That's actually a large rare disease at this point. Many of the rare diseases that we'll be talking about today are only dozens of people or hundreds of people. But this has been something that some would say is almost been a casualty of its own success, because so many companies ended up working in the rare disease space that in some cases they ignored some of the more common diseases. So some people criticize this for being actually overly successful. The priority review voucher programs, those have been put in place by Congress to help, again, get products into places where they might not otherwise have development. And the one that's relevant here for gene therapy is the Rare Pediatric Disease Voucher Program, because a gene therapy that addresses a rare pediatric disease, if it meets certain criteria, it can get a voucher. And as you may be aware, these priority review vouchers, they're essentially like negotiable instruments, and they convert a regular review at FDA to a priority review, and shaving 4 months off review time for a blockbuster drug converts into a lot of revenue. And so companies are interested in these, and they're negotiable. So if a small company gets one, they can sell it to a larger company that wants to use it. The expedited development programs include fast track, priority review, accelerated approval, breakthrough therapy designation, and regenerative medicine advanced therapy designation. Fast track, priority review, and accelerated approval have been around for a while. I'm not going to say a lot more about them, but I do want to at least give enough background on accelerated approval so that you can understand some of the differences that we have in regenerative medicine advanced therapy. All of these are for drugs or biologics that address serious conditions. We'll take it that the rare diseases that we're talking about here are going to be serious conditions. Now, normally accelerated approval allows us to use surrogate endpoints such as biomarkers or intermediate clinical endpoints that are reasonably likely to predict a clinical endpoint to give an initial approval with confirmatory trials that look at a more conventional endpoints on how a patient feels, functions, or survives. This is obviously a little bit controversial. You've heard a lot of back and forth about this because what you decide to use as that surrogate marker and how well that connects to clinical benefit can vary and reasonably likely to predict has some play in it. But this is what we work with. Breakthrough therapy designation is essentially an amalgam of fast track, priority review, accelerated approval use with additional agency input. And this was put in place in 2012 as part of the PDUFA reauthorization at that time. But as part of the 21st Century Cures Act in 2016, December 2016, regenerative medicine advanced therapy designation was put into place. And that was something that was put there because it was felt that cellular therapies, potentially including gene therapies, needed some extra oomph to get them across the finish line. And so the difference between a regenerative medicine advanced therapy designation and breakthrough therapy designation, aside from the fact that regenerative medicine advanced therapy designation is targeted at cells and potentially at gene therapies, is that the bar to get this is slightly lower than for breakthrough therapy designation. Breakthrough therapy designation, the requirement is that it has to the product has to be better than an existing standard of care, whereas for regenerative medicine advanced therapy designation, it only has to potentially address an unmet medical need. And then the designated products that get this have all of the standard breakthrough benefits, plus they have a different array of options that can be used to fulfill post-approval commitments if these products get accelerated approval. Real-world evidence can be used, expansion of follow-up time on the individuals enrolled in the initial trials can be used, and some other things. So it gives sponsors a little more leeway. But the real challenge to a lot of these therapies turns out not to be clinical development per se, although don't get me wrong, there are challenges in clinical development because when you have a very rare disease, it can be challenging to know what the baseline function of people are so that you can actually see if the therapy that you're giving is giving a big change in function. That's particularly true in neurodegenerative diseases where you have to try to see if you're making a change in a kind of a decline in somebody's function. Those can be challenging. But it turns out that one of the most challenging things for individualized gene therapies is actually the manufacturing. And that is because right now in the United States, the commercial viability in terms of what companies want to work with are generally gene therapies which treat at least 100 to 200 doses per year, because that, when you do a business model, makes up for return on investment for what goes into the manufacturing facility, et cetera, and yields a profit. The very large size here, the very large cup, that's for another day. Very large scale gene therapy production. We simply don't have the vector technology just quite yet for that, so we can leave that one alone. But the cup that's problematic is the small cup, which is, we have the technology to do in principle, but it is not cost effective. And because of that, some small batch gene therapies have essentially withered a bit. Now, you might say, well, is that really true in practice? Indeed, it is, because there are various gene therapies that have come on the market and have gone off the market in Europe because just the size of the populations are too small for commercial viability with our current model. And a lot of that ends up hinging upon the cost of manufacturing and the ability to manufacture in a facile manner different gene therapy vectors. It's possible, though, that I normally have several slides in between here, but I'll just, without going into the technical details, it turns out that a lot of the manufacturing of the most common gene therapy vectors, which are adeno-associated virus and lentivirus, can be automated, particularly for adeno-associated virus, which is adeno-associated virus is a cold virus that doesn't tend to cause any disease in humans. It turns out to be something that by and large also doesn't create any major adverse effects when people receive it. It's a little bit better in that way than adenovirus, which is a different virus. So there's adenovirus and adeno-associated virus. Adeno-associated virus has been used as a gene therapy vector because it turns out that it's a small virus. It can live without about 3,000 or so 3,500 base pairs of itself, and so we can stuff other things in there, such as the gene of interest, and that allows us to have these gene therapies produced. Now, it does create some challenges with larger proteins, but it's what has driven a lot of the gene therapy development. The other vector system, lentiviral vectors, are often used for chimeric antigen receptor T cells. They're used in vitro to change the cells, and that's easier. It's easier to use those lentiviruses, which are potentially integrating viruses for cells that aren't inside the body when they're actually manipulated. So the CAR T cells generally using lentiviruses. These gene therapies that we're talking about, the most common vector right now is adeno-associated virus. Those are by no means the only vectors that are used for each of these, but those are the most common. And it turns out that adeno-associated virus can be made using a more automated process, and we and others are working to try to do that, because you can imagine that if you didn't have to do things by hand, setting up everything from scratch, but you could move to a machine type of or a device type of production, that might make things a lot easier and decrease cost. And you can imagine then that you could have companies that would be doing this, and they'd have a portfolio of products potentially that they would manufacture and potentially have commercial viability. Other things we're trying to do to get towards commercial viability is to try to standardize what is done in the development and manufacturing of these bespoke therapeutics. A lot of the innovation in this area comes out of academic laboratories. And as someone who worked in one for a number of years and had one, the issue is that when you work in an academic laboratory, your idea is to get results, not to get standardization. And you often don't have the benefit of having a cookbook or a playbook. And so you just use what others around you have used or what you can get your hands on in terms of how you go about manufacturing or developing something. That's great if you're just going to do one thing, but when you have a whole ecosystem of people doing this, and you have 57 different varieties of things that are being transferred to, for instance, a contract manufacturer, the contract manufacturer each time has to rework these things. That adds cost. And so ultimately for the ecosystem, if we can start to have people agree and do things similarly, that may help expedite the development of these products. It'll help with technology transfer, probably also bring down cost. The other piece that we can imagine doing is the really cool thing about gene therapies is that unlike small molecules, which are, each one is got its own uniqueness, and it's true, some small molecules have the same, same side groups, things like this, or core chemical group. Gene therapies, when they're made in a given adeno-associated virus type, share the backbone of the gene therapy. And that backbone of the gene therapy is essentially what carries the part that's going to do the work, the insert that expresses the protein that's needed. And that backbone does not necessarily have to change from time to time. And if you can keep it the same, the idea is that potentially you could move from one gene therapy to another and not have to rework the entire vector up each time in terms of the manufacturing information, toxicology information. And that is kind of a razor blades analogy. The idea is that the gene therapy vector is the razor handle, and you can put different types of blades on it in order to go about your business. And that's the idea here. So just to summarize, we at FDA are very committed to advancing the development of therapies for rare diseases. I think we really want to help continue to help move forward product development, overcome limitations in manufacturing. We have labs that are working towards this, and we have grants that are going out to people trying to help in this regard. And we're also very interested in trying to make the clinical development as expeditious as it can be by looking at novel endpoints and novel trial designs. So I will stop there, and I think we even have a couple minutes for questions if I got it.
Great. We actually have 2 questions now, and so we'll ask Amy Rickheim. Okay, while Amy gets her situation together, Hawken Miller, introduce yourself and ask your question.
Worst comes to worst, why don't you just type it in the chat if you can?
That might be the way we do this. I will ask you a question while we wait for something to appear in the chat. Several of the conversations that we've had over the past couple of days have dealt with this issue of capacity building. Obviously, we have a very big international cohort. And so a lot of the conversations that we're having around access and collaboration may not apply in rural Ghana or in Malaysia. And so one of the questions that I had about FDA is, I wonder what is the scope of capacity building, outreach, programs, connection, what's going on in that regard?
Thank you. That's a really great question. We are very much involved in trying to work to build global capacity in this regard. In fact, we are we're meeting next week with the part of World Health Organization on a kind of a convergence paper on cell, tissue, and gene therapy. The issue here and it's really relevant here, is that in the United States, we might have 20 people with a rare disorder, but Europe might have 30 people with that rare disorder. Africa might have 50 people with that rare disorder. Asia might have 100. If you put them together, it's no longer quite as small a population. And you might say, well, it fits right into that commercial viability again. And granted, there are some issues around who's going to pay for it, et cetera. But there's this feeling that if we could come together as regulators and kind of agree for populations of certain sizes, that we would agree to accept similar information for these gene therapies, we might be able to have all of our populations better served, and particularly also then be able to better serve low and middle income countries, where sometimes gene therapies may be the best possible option for those individuals. I say that because if you think about somebody with thalassemia, sickle cell disease, or hemophilia that's located where there's no supportive care, those individuals have, although you might say, well, those are treatable. Yes, in a country that's a high income country, they're treatable diseases and we have good treatments for them. But in places where you don't have that kind of supportive care, the only way those people are going to really get treated long term is through gene therapy. And so we really, I think, have kind of this impetus that we really feel compelled to try to move this forward for the good of global public health. And I think ultimately it's mutually beneficial.
Hawken has typed his question, and so I'll ask it for him. He says his mother has actually worked with you through Cure Duchenne, and he wanted to ask where can expanded access and right to try be improved? He's heard that because expanded access drugs can't be charged by pharma companies, it's not common for them to use this pathway.
So I think I'm going to focus on expanded access. And the truth is that there actually is, if you look up if you can just search IND charging guidance, there is actually a way that that sponsors can recoup their costs. They can't make a profit, but they can have a certified public accountant basically vouch for the fact that they've spent X number of dollars making a therapy, so that if they make, as long as they've gotten permission from FDA, if they make a therapy, for instance, an example of this, I think in the Duchenne's area, is that there's a group of boys that are getting treated with gene therapies now. There are some older teenagers and young adults that are not eligible for those trials. And the question is, could you potentially see products being made available there? Right now, sponsors, they're very expensive, so they might not want to make them available, but they could potentially, in this case, if they've shown in the boys that there's some evidence that they might have benefit, they could potentially apply to FDA, and we potentially could allow them to have what's called cost recovery, which is that they can essentially charge back what it costs them to produce the product. And that's something that some sponsors take advantage of in various settings. So there are ways to make that happen. I actually think that a lot of this has to do with continuing us at FDA acknowledging to sponsors that we're not going to have adverse events in sicker patients potentially adversely affect the approval that they might get in their target population. That's one of the largest issues that we hear from sponsors. They're panicked that somehow if they give the product to an older individual who's not in their target population and there's an adverse event, it's going to besmirch their product that's being developed. And the truth is that it's extremely rare that anything like that happens. And when it does happen, it's because the adverse event that occurred in that older population is also occurring in the younger population. And by the way, that's a major problem for the whole product, as you know, it's across the whole product. So we try to do the right thing.
One of our speakers yesterday is actually responsible for me reaching out to you, and that's Emil Kakis, because he said, if you want to talk to the smartest people in the business, and one of the most interesting conversations he had recently was with you. And so I'm going to pull some of the things you just said and connect it with his main position, which is that the exponential growth that we've seen in screening may be hard to replicate when it comes to developing treatments because of things like commercially viable and adverse events, fear of adverse events. So in other words, we've gone from 32 diseases or 61 diseases that we knew 32 years ago to 6,000 now because of screening, but we still have these intractable barriers to developing treatments. So what is your response to this fact that the elephant in the room is that these barriers are going to make it impossible to achieve?
Yeah, so I think Emil's point is well taken, that we have to do what we can to start to bring down barriers, while at the same time, I talked about this balance. We want to bring down barriers while at the same time making sure that we don't degrade the system so that what goes out there is junk, right? So there is that balance, and I'm sorry to use the technical terms here. But the issue here is, I do think there are ways that we can probably lean in more. I think some of that is taking a little bit more chance on using our accelerated approval provisions by using surrogate endpoints for some of these diseases. Some of these things take enough years to figure out from clinical endpoints that they're going to actually work or not, that if we use surrogate endpoints, we could probably get through more diseases more rapidly. I think by using this concept of reusing information from one gene therapy to another in terms of the information about the vector. That will help get us there. And by essentially leapfrogging, moving forward the manufacturing and helping that to move forward. And that's starting to move now some. That may help as well. But it's a combination of those things that need to happen, as well as then also preparing the field to understand that with more of these, we'll have to figure out some way to pay for them as well.
A question from the other session, and I'm remembering it was about something, I think, GoFundMe movement in terms of therapies.
Yeah,
Can you tell us about that?
So we see some people trying to make gene therapies for rare diseases by GoFundMe campaigns. And as much as I think that, I'm not going to say there's anything wrong with that, of course, but for me, I just would like to see us find a way to make sure that all patients with rare diseases could get access to gene therapies and not just the more articulate. You know what I'm saying? It seems like there will be, if we rely on that as the method for funding here, we will end up with inequality in access. And it turns out that commercial production and payment from insurers is one way of helping to ensure, and at least in our capitalist system, that's one way of helping to ensure reasonable access. There's still inequalities. Don't get me wrong. I'm realistic there. I mean, there's still inequality there, but there's probably less inequality than when we have GoFundMe campaigns, when it tends to be more articulate individuals who are able to or more tech savvy that are able to make those things happen.
Are there any other questions among the fellows? If not, I'm going to jump in. Here's Hawken. Go ahead and ask your question, Hawken.
Dr. Marks, I wanted to ask you about gene therapy. You know, I know more about this just because of Duchenne and what's being done there, but you know, there's some issues with AAV antibodies and delivering a full gene. My general question is: gene therapy kind of the end all be all for rare diseases, or are there other methods to find curative treatments that you're looking at? And then the second question is, how are you looking to address some of those gaps with gene therapy?
No, great question. There are some rare disorders where the solution may not be a gene therapy. It may be a small molecule drug that can equally, when taken, can alleviate the disorder. It's just that the most straightforward way we can find things often today for certain rare diseases is actually gene therapy, because it turns out finding like a small molecule drug to do something requires screening, lots of compounds, and actually a drug development program that can cost incredible amounts of money. Whereas gene therapy, because you know what you're looking for, is sometimes more straightforward. It is true that AAV is not perhaps the most perfect of platforms, because some people have antibodies, and once you've been treated with an AAV, you generally develop antibodies to that, so you can't get redosed with it. So then if you want to redose, you have to use a different AAV type. And so people are working on better non-viral vectors. And if I had to look 10 years in the future, I would say we'll probably be starting to see an evolution from AAV to probably other non-viral vectors that have better characteristics. But again, I think it just so happens that gene therapies for rare diseases are a relatively straightforward way of getting there, but they're certainly not the only way of getting there. Obviously, for lysosomal storage disorders, many people have been treated, for instance, with Gaucher's disease, with enzyme replacement therapies, or for that matter, with small molecules you've probably heard about. And it's just that those were large enough rare diseases that it made sense to do that. For very small rare diseases, populations of 10, 20 people, the idea of trying to develop a small molecule or protein therapy is just often, it's often much more complicated than to just develop a gene therapy.
Thank you.
Our last question will go to Shao Chin Tan from Malaysia.
I would like to ask, as we know, and I think a few speakers have brought this up, because of the few numbers of certain rare disease patients, there might not be enough statistical power when it comes to clinical trials. So yes, how does the FDA accommodate that?
That's a great question. So something I didn't talk about is we're very interested in use of, for those type of situations, we feel like we can use things like Bayesian designs where each patient counts in trying to figure out whether something is actually working. So the first person that enters a trial, they could respond or they don't respond. If they don't respond, it means the next person is less likely to respond. If they do respond, it means the next person is more likely to respond. And the idea here is that if you do have a very effective therapy, you know, we've seen gene therapies where you actually are pretty sure that the thing works after 5 or 10 patients. So I think we potentially can do this, but we have to use different types of statistical models than we're used to. It still can be challenging. I'm not saying it's always easy, but we're beginning to accommodate these through the use of kind of alternative statistical designs.
Bijal, I think I can answer this last question before we let Dr. Marks go, and that is about how close we are to having a gene therapy for sickle cell. And our speaker tomorrow, Rob Stein, the senior editor at NPR, will be talking about some stories he did about the first patient in the US to be treated with CRISPR gene therapy, a woman in Mississippi, I think. Yeah. So I think we'll get the answer to that question and get more insights from Rob tomorrow, Bijal. But in the meantime,
you'll have a good time listening to that, because the issue of sickle cell disease and gene therapy it brings out some of the nuance and complexities and some of the tremendous promise in gene therapy. So it's a great topic.
Wonderful. So thank you so much for taking out time from your busy schedule to come and talk with us. And we hope we can stay in touch with your office for more information.
Certainly, please do. Thanks a lot. Take care.
Take care.
Bye-bye.